Hello, I am Professor Sims, and this video is about DNA structure, function and replication, RNA transcription, and protein translation. The learning objectives for this unit include describing the historical events that led to the characterization of DNA and heredity, describing the structures of deoxyribonucleotides and ribonucleotides. We'll talk about the similarities and differences between RNA and DNA, the three main types of RNA that are used in protein synthesis. I will help you to define what a gene is, the genotype, differentiate genotype from phenotype. We'll talk about chromosome structures and packaging, the processes of DNA replication, RNA transcription, and protein translation in prokaryotes and in eukaryotes. DNA was characterized long before its role in heredity was understood. Way back in the 1850s and 60s, Gregor Mendel was doing his experiments with pea plants so that he was able to figure out that phenotypes were linked to genetic information because he could crossbreed different pea plants and see that dominant characteristics were being passed on, recessive characteristics were not. That's all you need to know for this class. Mendel will be the subject of much discussion if you are taking genetics. In 1869, Friedrich Miescher, he was able to isolate this stuff. It was nasty looking, it was full of phosphorus and proteins and stuff, and he got it from white blood cells. And a really interesting thing about that is how he got the white blood cells. He actually was collecting medical waste, like pus from bandages and stuff. But he was able to purify this stuff, and he called it nuclein. So that's way back in 1869. Some years later, Albrecht Kossel, he actually was able to characterize specifically the nucleotide bases that were found in this nuclein, and it was now called nucleic acid, and it got the acid part because of its low pH. And then using it's an algae, I believe, Acetobularia, Joachim Hammerling, he was able to demonstrate that the nucleus is where hereditary information is found in the cell. And this is figure 10.3. These were some of the experiments that he did where he was able to determine that. What's really funny about this is an algal cell, and it's actually got its nucleus down here in the foot. So the nucleus is here, and it grows this stalk, and it has the cap. This is how it reproduces. But what he did was he cut the cap off, and it grew a new one. But if he cut the foot off, nope, it died, right? No new foot was regenerated. So that's how he determined that the hereditary information, what we now know as the nucleus, was actually in the foot of these organisms. And then in a second set of experiments, he was able to, so he would take an intact organism and he would cut off the cap, and then he would take another species or similar species that had a different phenotype and graft it onto the foot. And then sometime later, as long as this nucleus was intact, right? He didn't cut off the foot, he cut off the cap. And he grafted it onto the foot, and it still made a cap just like it normally would. And the other species, he did the same thing with it and grafted. So he was basically doing cross hybridization experiments here. And it was determined that, whichever species the foot came from, that's the type of cap that it grew. In 1928, Griffith, he was able to show that a dead bacteria that had capsules was able to pass on genetic information to live, non-encapsulated bacteria. And then a little later, in 1944, Oswald Avery and Colin McLeod and Macelin McCarty, they actually identified the compound that was in these bacteria as being DNA. DNA stores genetic information, and this was unequivocally demonstrated by Alfred Hershey and Martha Chase, as published in 1952. So what they did was they labeled DNA from viruses that were infecting bacteria, and the labeled DNA that came from the virus was identified inside of the bacteria. And they also were able to determine that the protein coats in the virus, the capsid, had nothing to do with this transmission of DNA. This is figure 10.9 that is describing these experiments that they did. So they have a radioactively labeled viral DNA, and then it attaches and infects bacterial cells. And then they cultured those bacterial cells out, and they were able to identify that viral DNA in the bacterial cell. This is a timeline of some of these discoveries, and it goes beyond what we talked about already, but we'll get to those people. So Mendel was very early on, and then Griffin, his experiments here. We didn't talk about all of the, you don't really necessarily need to know every single one of these names and dates, but you should know the ones that we just talked about in the presentation, and there's going to be some more like Shagraf, Watson, and Crick. We'll be talking about those in just a bit. But this does give kind of an idea. So from 1865 all the way up until finally in the 1990s, we were discovering what DNA was, what it was used for, how it was transferred, how it's replicated, what its structure is, what its function is, and then now we actually can map the genomes, the entire genomes, the entire DNA sequence of organisms. So nucleic acids are composed of nucleotides. Nucleic acids are the units of DNA, and nucleotides, they all contain a sugar, a phosphate, and a nitrogen. So a pentose sugar, pentose means it has 5 carbons, a phosphate group, and a nitrogenous base, deoxyribonucleotides. This means that the sugar, the pentose sugar that's in it, is actually a deoxyribose. And then pyrimidines and purines are the types of nitrogenous bases. Pyrimidines have they're a smaller structure, and they consist of either cytosine or thymine. The purines are the ones that have a larger structure. So the bigger word is the bases that have smaller structures. The smaller word is the bases that have larger structures. The purines are your adenine and guanine. And these things are linked together in DNA via phosphodiester bonds. There is, on the 5' end, there's a phosphate group. On the 3' end is a hydroxyl group. And then the whole nucleic acid strand, it has a free phosphate group at the 5' end, right? At the 5' end is a free phosphate group, and at the 3' end is a free hydroxyl group. And that becomes important when you start talking about DNA replication, because these are where things, the hydroxyl group, right? This is where new nucleotides can add to the growing strand of DNA as it's being replicated. But I'm getting ahead of myself. Let's look at figure 10.11. This is showing the structure of a deoxyribonucleotide. So here, when we're talking about phosphate groups, this is the phosphate group. Notice that it does have a negative charge on it. This is the sugar, and then this is the nitrogenous base. And these are this right here is what we're talking about when we're talking about the pyrimidines and the purines. So this base here is either going to be adenine, thymine, cytosine, or guanine. And this is just a more specific structure of the sugar group right here, and it's showing where it gets this 3', 5'. It has to do with its stereochemistry in the way that it's named. This is carbon 1, carbon 2, carbon 3, carbon 4, and then carbon 5. Carbon 5 is where the phosphate group is attached. Carbon 3 is where the hydroxide or the hydroxyl group is attached. These are the structures of the pyrimidines versus the purines. Notice the pyrimidines have a smaller structure. It's just one carbon ring with groups attached to it. And the purines, the shorter name, have the larger structure. They all have 2 carbon rings with groups attached to it. And this is important because of the way that they fit together. Adenine only binds with thymine. Guanine only binds with cytosine. And it's due to their stereochemistry. The nucleotides are linked together in DNA via phosphodiester bond. Figure 10.13 is showing how the phosphodiester bonds are formed and how the nucleotides are attached together. The phosphate groups are on the outside of the DNA molecule, and the nucleotides are in the inside of the DNA molecule. The nucleotides are held together to each other on the inside of the molecule with hydrogen bonds. And that is important because hydrogen bonds are not as strong as covalent bonds or even ionic bonds, which means that they can be broken apart relatively easily. That is, the hydrogen bonds can be broken apart much more easily than the phosphodiester bonds that hold the backbone together. But again, I'm getting ahead of myself. Let's talk about Chargaff. He was the person who was able to determine that only adenine binds with thymine and only guanine binds to cytosine. This is now known as complementary base pairing. Watson and Crick are famous for determining the structure of DNA, but it was Watson and Crick who are famous for determining the structure. But it's important to remember that they built upon the work of lots and lots of people that came before them. Essentially, the DNA is two anti-parallel strands that are bound together at the nucleotides in the middle via the hydrogen bond. Breaking of those hydrogen bonds can be accomplished using heat or chemicals, which is known as denaturing the DNA. If you're using heat to break the strands apart, subsequently cooling them will make them come back together. If you're using chemicals, then removal of those chemicals will bring the DNA molecules back together as well. DNA includes what amounts to the blueprints for building and controlling the cells in living organisms. These blueprints are heritable via vertical gene transfer. Vertical gene transfer is the term used to describe DNA, which is transferred from parents to their offspring. Figure 1016 is showing the overall structure of DNA. DNA is a double helix with two individual strands. Here and here is the phosphate sugar backbone, and then these guys in the middle are your nucleotides that are held together with hydrogen bonds. Remember that adenine only binds with thymine and cytosine only binds with guanine. Figure 1014 shows the famous photo 51 taken by Rosalind Franklin in 1952. This is an X-ray diffraction pattern that revealed the double helical structure of DNA for the very first time. Here is a short video further illustrating the structure of DNA.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Erwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel.
DNA, also known as deoxyribonucleic acid, and RNA, aka ribonucleic acid, are very similar molecules, but there are a few differences. First, while DNA is double-stranded, RNA is single-stranded. Next, the pentose sugar in RNA is not deoxyribose, as in DNA, rather it is ribose. And finally, RNA contains uracil instead of thymine. There are 3 types of RNA. These are your messenger RNA, ribosomal RNA, transfer RNA. So mRNA, rRNA, and tRNA. Messenger RNA is the intermediary between DNA and the synthesis of protein. Ribosomal RNA ensures proper alignment of amino acids and catalyzes the formation of peptide bonds during protein synthesis. And transfer RNA carries amino acids to the site of protein synthesis in the ribosome. Although RNA is not usually used to store genetic information, it can be used for this purpose in some species of viruses. These are the structures of deoxyribose versus ribose sugars and the thymine versus uracil nucleic acids. So notice the similarities. The main difference between deoxyribose and ribose is that the deoxyribose is missing an oxygen right here. Ribose has this OH group, and this one only has an H. And the main difference between thymine and uracil is shown here. Thymine has a CH group here, and uracil only has a CH here. So they are very minor differences. Structurally, they are very minor. A figure 1021 compares the overall structure of DNA versus RNA. So here's where they still look quite similar, but you can see that they are quite different. DNA is double-stranded and RNA is single-stranded. But while RNA is single-stranded, it can kind of fold over and fold itself in half. And in this way, it can still participate in complementary base pairing within the molecule, in which case uracil binds to adenine instead of thymine, right? Uracil adds to adenine and cytosine adds to guanine. This is an illustration of how mRNA, rRNA, and tRNA are used in protein synthesis. Remember that mRNA is the intermediary structure between encoding DNA and the resulting proteins. tRNA carries amino acids to the site of protein synthesis in the ribosome, and RNA helps align the amino acids and catalyzes the formation of peptide bonds in the growing chain of amino acids, which will become a protein. These structures and functions will become important when we discuss transcription and translation a little later in this presentation. The tRNA molecule is single-stranded, but it has this complementary base pairing where it's folded in on itself, and that's what gives it this 3-dimensional structure that looks like an upside down T. So this is tRNA. It's a single strand that has folded in on itself several times, and it gives us this 3-dimensional structure. It's a bit ironic that it looks like a T because the T in tRNA actually stands for transfer RNA. Again, this figure will be significant in your understanding of transcription and translation, which is section 11.3 in the text. But for now, just notice the group, this little group down here of 3 bases. These are paired to complementary bases on the mRNA strand, and this is called a codon. It is integral in the synthesis of amino acids and proteins. A genome is the entire genetic component of a cell. This is the entire sequence of DNA for an individual organism. Genes within the genome encode for proteins, mRNA, rRNA, and tRNA. The genome describes the genes as individual. The genome describes the gene an individual possesses for a specific trait. Genes can be differentially expressed based on dominance versus recessiveness, or based on certain environmental conditions. A phenotype is different from a genotype because it is observable traits that result from gene expression. So these are things that you can see. For example, my phenotype for hair color is that I have brown hair. A phenotype can be altered based on the expression of genes. For example, a young individual may have a gene associated with a certain illness that will only be expressed later in their life. Most genetic material is organized into chromosomes. The number of chromosomes found in the nucleus or in the nucleoid in the case of prokaryotes is used to classify organisms. For example, most prokaryotes have one chromosome, which is called haploid, and many eukaryotes have more than one chromosome, usually two, and that is called diploid. What that means is their chromosomes come in pairs. They have multiple linear chromosomes in the nucleus. So they differ from prokaryotes because most prokaryotes only have a single circular chromosome in the nucleus or the nucleoid because, prokaryotes don't have nuclei, right? And the way that these really long, complicated, very complex molecules of DNA can fit into the nucleus or the nucleoid is through a process called supercoiling and DNA packaging. And this has to do with binding proteins that coil the DNA up together, and they only become uncoiled whenever the cells are undergoing division. So before DNA is replicated, before the cells divide, the chromosomes become uncoiled. Eukaryotes and archaea have histone proteins that do this, and bacteria use different kinds of proteins, but they have similar function. This is figure 1025 showing this is a diploid chromosome, and this is also showing that there are not only genes, right? So there's DNA in the chromosomes that code for genes, but there's also quite a bit of non-coding DNA that's included in the chromosomes. And this non-coding DNA, it's not very well understood exactly what it's for, but there are lots of studies out there that show that some of it is involved in gene expression, so turning on genes or turning them off. And also they help to maintain the structure of the chromosome. They might have something to do with the processes of supercoiling and packaging, DNA packaging. There is such a thing as extra chromosomal DNA. In eukaryotes, this is DNA in mitochondria and chloroplasts, which makes sense if you remember that those organelles are most likely of prokaryotic origin. So if you remember a couple of lessons ago, we talked about how mitochondria may have been first incorporated into eukaryotic cells via endocytosis. Extra chromosomal DNA in prokaryotes is usually in plasmids. So they'll have most of the DNA that's in the nucleoid, but then they may also have plasmids. And these plasmids are used to transfer genetic material from one cell to the other, and this is called horizontal gene transfer. So if you remember the hereditary DNA, like from a parent to an offspring, that's vertical gene transfer, but transferring DNA from one cell to another is considered horizontal gene transfer. So we're going to talk about DNA replication, and I actually want to show you another video. I'm sorry if these videos drive you crazy, but to me, I'm a very visual learner, so it helps. And I am trying to, and I have, throughout this entire course, trying to get you guys to see the same kind of concepts and things by audio, visual, and kinesthetic learning. So a lot of the kinesthetic stuff, the hands-on stuff, obviously, is why we have the lab course. But for these videos and the PowerPoints and the text, the reading, and even with your oral presentation, I'm really trying to kind of hit the audio and the visual part. So anyway, enough of my teaching style. Let's have a look first at the video, and then we'll go through it with the information from the text and the figures and things afterward.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment.
Okay, so the DNA that you start with, it's not destroyed in the process. And it is altered in such a way that it's broken apart and it's used as a template in order to synthesize new strands. And what you end up with is, if you start with one DNA molecule, at the end of replication, you'll have two identical DNA molecules. So first of all, the DNA in the chromosomes, it's super coiled. It's unwound usually by an enzyme called gyrase, and this is generally in eukaryotic organisms. What they don't have in here is topoisomerase. Topoisomerase is an enzyme that helps when the DNA is being uncoiled and stretched out. It helps to keep the phosphodiester bonds together. It keeps the sugar phosphate backbone from breaking. So that's topoisomerase. Helicase is the enzyme that breaks the hydrogen bonds between the bases. Adenine, thymine, guanine, and cytosine, right? They're held together by hydrogen bonds. Helicase breaks those bonds. Primase is the one that makes the RNA primer that attaches. And then DNA polymerase is the enzyme that adds new nucleotide bases. And then that process, adding the new nucleotides, that's called elongation. And the leading strand is the one that goes from 3' to 5', and that's where continuous replication occurs, and the lagging strand is the one going in the opposite antiparallel direction, and that's the one where Okazaki fragments are added. And ligase is the thing that it kind of comes and cleans everything up. It fills in any blank spaces between the Okazaki fragments. So this is what it looks like in figure 11.7. One of the reasons I wanted to show you that video is because this figure is just a whole lot to look at. These blue things, these big long blue things, are the parent strand or the template DNA, the DNA that you started with. Gyrase is the one that's responsible for uncoiling it, and then topoisomerase is the enzyme that helps keep the phosphodiester bonds held together. And then helicase is the enzyme that splits the strands apart by breaking those hydrogen bonds in the middle. DNA polymerase here. Here's the RNA primer. This is the lagging strand. This is the leading strand. DNA polymerase is adding new base pairs to the leading strand. And this DNA polymerase is adding new base pairs to the lagging strand. And then ligase is filling in those gaps in between the Okazaki fragments on the lagging strand. The next thing we're going to get into is RNA transcription. And in order to just kind of keep things straight here, this is an extremely simplified figure 11.2, but it is good to kind of have something really simple here to illustrate why we're talking about these things in the order that we're talking about them, okay? So the central dogma of biology is this, simply. DNA encodes messenger RNA, the mRNA, and then the mRNA is used to encode for protein. So DNA transcribes mRNA, and then the mRNA is translated into protein. So DNA replication, transcription, translation. So let's talk about transcription. In transcription, the genetic genetic information from the DNA is used to make RNA. RNA polymerase synthesizes RNA, kind of similarly to how DNA makes DNA, but by adding complementary RNA nucleotides to the 3' end of a DNA strand. RNA is using DNA as a template for making more RNA. It binds to the DNA at a sequence called a promoter during the initiation of transcription. Genes that encode proteins of related functions are frequently transcribed under the control of a single promoter in prokaryotes, and this is called polycistronic mRNA. RNA polymerase is similar to DNA polymerase, however, RNA polymerase does not require a free 3' hydroxyl group in order to add nucleotides. So it doesn't need a primer. There's 3 different RNA polymerases in eukaryotes. Eukaryotes do not have polycistronic mRNA, they have monocistronic mRNA. Eukaryotes only have a single polypeptide that their mRNA encodes for, which separates them from prokaryotes who do polycistronic mRNA. Eukaryotic primary transcripts are processed in several ways, including the addition of a 5' cap and a 3' poly A tail, as well as splicing in order to generate a mature mRNA molecule that can be transported out of the nucleus and is protected from degradation. So once one of these mRNA has been synthesized, okay, so if you're going from DNA to protein, mRNA is the intermediate step. It's the step in the middle, right? You have DNA replication, transcription, and then translation. So the transcript is the mRNA, the bit in the middle of this process. And it does, in fact, have to be moved out of the nucleus in order for it to be used to make proteins. So your biology has ways of protecting this mRNA in its journey. So I hope that kind of clears it up a little bit. Now the third step, right, in the process of making a protein from DNA is translation. So mRNA, right, is basically the substrate for this, and it includes some tRNAs. Remember, tRNAs are transfer RNAs, and they match different codons to amino acids, and that is what determines which proteins are made. There is a genetic code, and it's degenerate, and what that means is that several codons can code for the same amino acid. It's also pretty much universal amongst all living organisms, which is a huge deal. So the codons in my DNA, your DNA, are the same as those in, insects, algae, bacteria. It's pretty cool, actually. Ribosomes are composed of a large subunit and a small subunit, and we saw that already earlier in this presentation. And they have different sizes between the different groups, and each subunit in the ribosome contains a ribosomal RNA and a protein. This is figure 1112, and this is showing the genetic code for translating nucleotides that are in triplets. So they read them 3 at a time. These 3 nucleotides, so in this case it's uracil, 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 right? And they are in the mRNA that was synthesized from the DNA. You're not going to have T's in here, you're going to have U's. But 3 nucleotides together are codons. They code for an amino acid. And then the amino acid then codes for protein. This happens in different steps. Basically, there's 3 big steps for translation. You got initiation, elongation, and termination. So initiation is when the small ribosomal subunit binds with initiation factors. And then an initiator tRNA binds at the start of a codon, okay? And then there's this thing called an initiation complex that attaches to the large ribosomal subunit. So everything is coming together. That's initiation. Elongation, you have the tRNA binds directly to the mRNA in the A site of the ribosome. Then a peptide bond is catalyzed between the 2 adjacent amino acids, because remember, proteins are just long chains of amino acids, okay? So the ribosome moves 1 codon at a time along the mRNA. The whole process terminates when the ribosome encounters a stop codon. And a stop codon is a codon that doesn't code for anything. So the reaction essentially stops. Okay, so here back in 1112, these are examples of stop codons. Okay, so when the ribosome encounters one of those codons, it terminates translation. So this is figure 1115, okay, and it's showing the different roles of the small and large ribosomal subunit. This here is the mRNA. It starts when the tRNA brings the first amino acid into the polypeptide chain. Then elongation is amino acids are being synthesized one by one. It goes down one codon at a time. And as the amino acids are being synthesized, they're being added to this chain. The chain eventually will become, a protein. And then the termination is when the ribosome encounters a stop codon.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA. In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
That is going to do it for Lesson 9 material. As always, thank you guys for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.